Hello, my name is Dan Tempest, and today I am going to present to you on muscle structure and how that relates to the contraction of a muscle. All right, so this functional unit of a muscle contraction is going to be the sarcomere. Essentially, that's going to be a muscle cell, and you can see the breakdown of how that works. So we have the bicep muscle here, and they're going to break it down into a bunch of bundle of muscle fibers, and then here we have one sarcomere, which is going to be within a muscle cell here. And you'll notice that there's some light bands along with the Z line, along with the dark bands. And I'll go through what each of those do. But essentially, we have alternating bands of thin and thick filaments. You can see the dark band in an actual microscopic image here. You'll see the dark bands here and the light bands with the Z lines right here. Okay. So the structure of a muscle fiber, you can see again here we have the dark band, the light band with a Z line, a Z disc. And within there, we're going to have a sarcoplasmic reticulum, which is going to store your calcium. And I'll go more into that on the next slide. Here you can see the nucleus of the cell. Muscle cells have tons and tons of mitochondria, so I'll point out a couple of those there's one labeled here as well. The sarcolemma would be what wraps around that then. All right, so the sarcoplasm is essentially the muscle cell's cytoplasm, and then again, this is going to contain many mitochondria. Very, very important that a muscle cell has a lot of mitochondria. Mitochondria's chief job is to create ATP through cellular respiration. And because a muscle cell's job is to contract, it requires a lot of energy in order to be able to do that. So the mitochondria functions to create a lot of that energy, allowing that muscle cell to continue its work. Its cellular work is what ATP essentially's job is to allow for. So ATP is what is created by the mitochondria through cellular respiration. ATP allows the muscle cell to do its work. Sarcoplasmic reticulum is another really key. Essentially, this is similar to the endoplasmic reticulum within any cell. And its main job within a muscle is to store calcium. That calcium is going to be released eventually through an action potential that would stimulate that. And calcium eventually be pumped back into that sarcoplasmic reticulum in order to allow the muscle to eventually relax, and we'll go into that. But a sarcoplasmic reticulum is essential to the functioning of any muscle cell. All right, so here we have the actin and myosin. So within a muscle fiber in a sarcomere, you have interacting proteins. Those proteins are actin and myosin. So the thin filaments, which are these darker blackish colored on here, almost looks like a backwards E or a 3 here, on that, depending on what side you're looking at. So those thin filaments are 2 strands of actin and 1 of tropomyosin. There's also some troponin on there as well that would move the tropomyosin when the muscle is stimulated. We'll go into that a little bit more later on in this presentation. Then you have the thick filaments, which are composed of the myosin molecules with their globular heads. Those globular heads will interact with the actin during the cross bridge, and we'll go into within a contraction. Okay. Oh, sorry. I'm going to go back here. So again, actin, it's going to be essentially a braid of actin and then the tropomyosin molecules. And there's going to be troponin molecules that are going to move the tropomyosin at certain times, allowing for cross bridges to be achieved. Within the thick filaments, again, we have the myosin, that we have that molecule with a long protein with a globular head on the end. You can see some of those heads within this picture. All right. So when a muscle cell is triggered, this would be done by a motor neuron. So the motor neuron is going to stimulate. You can see an action potential coming down this motor neuron. It's going to stimulate and then release acetylcholine across the neuromuscular junction. That's going to trigger an action potential actually within the muscle, and that's going to trigger calcium release from the sarcoplasmic reticulum. So the action potential stimulates that muscle cell sarcoplasmic reticulum, which releases the stored calcium. When a muscle cell is at rest, the tropomyosin is going to block the actin binding sites where the myosin heads would bind to that actin. So those are blocked when the muscle is at rest. Calcium will trigger troponin, to move those tropomyosin braids and expose those heads. And I'll show you a picture that shows that really well in the coming slides. But the muscle has to be triggered by a motor neuron in order for any of this to occur. So here we have at rest. You can see here we have the tropomyosin blocking. You can see these darker binding sites. Okay, so these are all blocked. And when calcium binds to troponin, those will be now exposed. So now you can see down here on the bottom picture, now those myosin binding sites on the actin are exposed. So calcium is what triggers this. Troponin is what actually moves the tropomyosin. Again, that will expose those binding sites. When that happens, you can see down here, again, we have up here would be at rest. Tropomyosin is blocking those sites. Calcium binds. You can see the calcium is now bind to the troponin, and that's going to expose our binding site. So you can see here now a myosin globular head is going to interact with that binding site. And when that happens, that myosin head is going to contract or bend and going to allow that muscle, that sarcomere, to contract. Okay, so again, it's the calcium that allows the tropomyosin or the troponin to move, and the tropomyosin exposing those heads. Then the myosin globular heads can expose and move eventually that actin inward. So if we look here we have, again, our purplish is going to be your myosin. Here we have the actin, and as you see, this is the start. And if we compare that to the muscle contracting, eventually gets contracted fully, you can see those actin heads have actually even slid past each other. So the Z line, essentially those Z lines are going to move closer together, and that muscle will contract. The muscle will need ATP. So the ATP, actually, if I go back, you can see this myosin globule head has contracted, or it has bent. In order to get it back so it can bind to a new actin binding site, it needs ATP to reset itself, essentially. So ATP is very important in allowing that contraction to continue so that those myosin heads can continue to go back and rebind. And that would be what we call the sliding filament theory. So that sliding actin filament is going to be continuously slid by those moving globular heads. So here we can kind of show you that, actually. So that ATP is going to allow that globular head to move back. And here you can see it's going to bend forward, allowing for that muscle to continue. But what you're seeing here, if I go through this, we have your motor neuron up here releasing acetylcholine across that synaptic cleft. So this would be your neuromuscular junction, and the action potential is then propagated within the muscle cell itself. So here we still have an action potential. The action potential within the muscle itself is going to cause calcium release from the sarcoplasmic reticulum. That calcium will then, see here we have it flooding into the sarcomere. It's going to bind to troponin. Troponin is this purple molecule that's bind to the tropomyosin. When that happens, the tropomyosin moves slightly, and that's going to expose our actin binding sites that where the myosin heads can bind. Okay, so that calcium triggered the troponin to move the tropomyosin. Now the globular head can connect, as you see here, and it's going to bend and contract that. Then ATP is going to use to reset that myosin head, and it can continue to contract. Eventually, in order for the muscle to relax, the calcium has to be pumped back into the sarcoplasmic reticulum. And when that fully happens, the muscle can relax into its relaxed state. But during the contraction itself, that calcium needs to be flooding that muscle cell so that the troponin can move the tropomyosin, releasing or exposing the binding sites. The myosin globular heads then are going to use the sliding filament theory and continuously contract. So that's what you're seeing in this picture. And then just go through my references. I used my AP Biology textbook, specifically the unit on the muscular system. That's where most of the pictures came from, and they are cited on each slide. I did also use a crash course video on muscle cells to refresh me. It's been a while since I taught the muscular system. So I used that to refresh some of my memory. And then I also used our class textbook, the muscle physiology section of that. So I hope you enjoyed. That's how the muscle structure aids in contraction of a muscle. Take care.